Thank you guys for coming. My name is Ira Clark. I'm here at UCLA. I'm assistant adjunct professor and director of biomedical research minor. And my colleagues here are.
I'm Alexandra Tower. I'm at Santa Monica College in the Life Sciences Department.
I'm Casey Shapiro, and I work in the assessment group in our Teaching and Learning Center here at UCLA.
And so we'd like to tell you about our project we've been running on research deconstruction and maybe kind of provide some tools for learning how to incorporate this into your own classes. So this is a project we've been running for the last few years with support by the California Education Learning Lab.
We have partners at, I'll go back to the slide.
Yes.
In addition to our colleague Alex, who's here from SMC, we also have colleagues who are using our modules at Fresno State as well as Glendale Community College. So we're very thankful to have the opportunity to really test out the deconstruction both at Cal State and at a community college. So.
Yeah, actually,
sure.
The first few.
Yeah, absolutely. So I think it's important for us to know a little bit about you. So just kind of want to see who's in the room so that we can try to give information that's more tailored to your own campuses and your own interests. If you would, raise your hand if with any of the categories that you identify with. So how many here work at a community college or teach at a community college?
Oh.
Teach at a four-year institution. Hold an administrative role at your institution. Have been teaching for more than 10 years. Have been teaching for less than 3 years. Interested in collaborating with your colleagues at other institutions. Teach research methods and procedures in your introductory courses. Oh, great, welcome. Teach courses that enroll in more than 100 students. And then finally, teach STEM courses to students majoring outside of STEM, so like a non-majors course. Well, we think that the work that we're doing with generous funding from the Learning Lab will hopefully be something that you're interested in for teaching your major classes, your non-majors classes, or chemistry class. So we have a, oops, did I go the wrong way? Yes. Here's a quick agenda. Just want to do the introductions. We're going to go through what research deconstruction is, and then show the example of one of our modules that is for our majors biology course. Then we're going to go through the toolkit that we developed so that you can see how this might fit into your own class. And then do a deeper dive and again, like how you could use this in your own courses. And then as the assessment person on the team, I'm going to show you some of the work that we've done to collect information from our participants, both the instructors who have implemented as well as students who have been part of our deconstruction courses. And then we'll end it with time for questions.
All right, well, thank you. So, so what is research deconstruction? This is a model we developed nearly 20 years ago now. It was developed together with Rafael Romero and it was called Banerjee. And it is a way of introducing students to the process of scientific research and discovery and scientific inquiry through the unpacking of a research seminar. So the way this works is that we, first of all, I should say we have no prerequisites for this class. So we teach this to students who are first-year students. They've taken no introductory biology, as well as more advanced students. In our classes at UCLA, we have up to 90 students in a class, but it can go higher than that if you've got TA support. So it's pretty scalable. What we do is we invite a faculty speaker or a senior scientist to deliver a PhD level research seminar. That seminar is well beyond the understanding of any of the students in the class. It actually probably reinforces all of their feelings of imposter syndrome and, research is something that's beyond me. We've warned them in advance that they're not likely to understand much of this. It's okay. Just get what you can out of it. We record the seminar, make it available to the students throughout the course so they can review it whenever they want. But then the instructor of the course, who's not the invited speaker, unpacks that seminar over a series of deconstruction classes. So in unpacking the seminar and deconstructing it, we focus first on what's the big picture questions of the research. Maybe it's understanding the brain, maybe it's understanding how cancer develops, maybe it's understanding basic mechanisms of cell division or gene expression. We go through a little bit of background science for a couple of classes. Then we dive into the specific aims that were presented in that seminar, some of the experiments that were presented to address those aims, and then which means we're getting into experimental techniques. And then we talk about the data. How do you interpret that data? Is your interpretation different from what the speaker gave you, for instance? This unpacking in our class at UCLA, which is an elective class that's just devoted to research deconstruction, takes about 7 classes for one seminar. So we're doing this very slowly. We start off with a take-home reflection assignment where the students just identify what they think the big picture questions are. What do they think was the take-home message, the conclusion? What model systems were used? What did they find to be the most exciting or interesting result that was presented? And what did they find most confusing? And we tell them this is just graded on effort. We haven't deconstructed anything yet, so we don't expect any kind of deep understanding of the seminar. Answer all 5 questions, you'll get full credit. All right, it's not based on accuracy. But it provides the student with an opportunity to think about what did they get out of the seminar. It also provides us as instructors an opportunity to see what the students are getting and where they might need the most help. And then, and this is something that kind of came out of this project, we now actually have the students do this assignment again at the end of the deconstruction to reflect and see how much they've learned. It's important in doing a deconstruction, I think, to deliver content as it's needed. So you don't explain all of the techniques that were used, for instance, at the beginning, and then expect people to remember it 3 classes from now when you're actually talking about the experiments. You explain techniques and concepts that are necessary as they come up. And you want to lead your students through the story. So remember, the whole point of this is for students to understand how scientific discovery is made. You essentially want them to kind of experience vicariously the process of asking questions and then figuring out ways to answer those questions through experiments. Now, the other thing that we do, which I'm not indicating here, is that at UCLA we also will regularly have a Q&A session at the end. So after we've gone through 7 of these deconstruction classes, we'll bring the speaker back for an hour-long Q&A session, and the students then have the opportunity to ask questions of the speaker, and we ask every student to write up a couple of questions that they might want to ask the speaker and turn those in. So every student in the class gets to practice asking questions, which I think is an important part of game science. All right, so we've been assessing this for years. We've seen evidence through the CURE survey that David Lopato administers that this provides students with what they feel is a better understanding of how scientists approach problems. We've also seen some evidence that this might be improving STEM persistence and STEM retention. And so we wanted to know, can this have broader utility? And one of the things that we realized with some of our early efforts in exporting, for instance, to Santa Monica, was that there's little reason for students to take a purely elective course. There's little reason, if it's a purely elective course, it's not even articulating to a 4-year college, there's no motivation to borrow from my previous session, there's no motivation to get the buy-in from either students or for faculty and departments to develop a brand new course. And so we wanted to know, can we actually incorporate this into existing courses? And this is where our Learning Lab project kind of came in. We wanted to know, could we build this into the introductory biology and chemistry courses, and also general biology courses that that students are already taking. The intro bio and chemistry courses are often gateway courses for students pursuing STEM majors. Chemistry actually came out of the interest that Fresno State had in developing this with some of their chemistry faculty. We had never even done chemistry research deconstruction at UCLA. So it was a really exciting thing to try. So our lead partners at the campuses were Alex Tower here at Santa Monica College, and then Shelley Tai at Glendale Community College, and I am not good at this laser pointer, and Chris Meyer at Fresno State. All right, and so I'm just going to show you some components so what we did was we developed modules for introductory biology, introductory chemistry, and general biology. Each module begins with a pre-recorded research seminar that we recorded here in our studio at UCLA. And so for the introductory biology, we invited Stephanie Correa, who is faculty in integrative biology and physiology here at UCLA, and she presented a seminar on how estrogen regulates temperature in mammals. So these were mouse experiments, but basically the idea was that estrogen signals to a subset of neurons in a region of the brain called the hypothalamus, and then those neurons somehow change their gene expression and somehow contribute to altering temperature, core body temperature. All right. And so in deconstructing this work, you actually get to touch on a number of topics that some of which are in the intro bio syllabus. So homeostasis, cell signaling, sexual dimorphism, gene expression, how gene expression contributes to cell identity, and then some more sophisticated things like how do we detect gene expression? How do we interfere with it? Right. So one of the things that we also hear from faculty is that there's just so much you have to cover in a syllabus, and where are you going to find the time to start to unpack some of these experimental techniques? So to help with that, we also recorded a whole series of video tutorials. So many of these were produced by my colleague Rafael Romero for the introductory biology module. And so these were tutorials that students could watch before class, and then the instructor in kind of a flipped class format would review some of the concepts or material from, say, a video on immunostaining, so that you don't have to completely explain immunostaining. You can review that, but now use a piece of the seminar to see how is immunostaining actually used in one of the experiments from the seminar. Okay, so it was a way of kind of saving a little bit of time, in class time for deconstruction. So as I said, we did this not only for introductory biology, but also for chemistry and for general biology. So our chemistry speaker was Jenny Yang from UC Irvine, and our general biology speaker was Elizabeth Torres from Cal State LA. So Jenny Yang was speaking about energy storage and carbon capture and using electrocatalysis to help make that happen, to improve energy storage and methods of carbon capture. Elizabeth Torres was talking about bioluminescence in marine organisms called ostracods. We produced a whole series of video tutorials for general biology as well. So Alex and her team at Santa Monica actually took the lead in that. In addition to these seminars, we also included a short 5 to 10 minute video by each speaker talking about their career path and how they got started in STEM. And I want to point out one other thing, which is that we were very intentional in choosing speakers who were really, successful scientists, and were not stereotypical depictions of scientists. We wanted, we were very intentional in choosing 3 women scientists, women of color. So some of these are, at least a couple are first generation college students who got involved in STEM and now are faculty at universities. And you know, running really exciting research programs. So I now want to just do a little bit of a deep dive into one of these. So we're going to do just a short dive into a piece of Stephanie Correa's seminar. And then we'll have some time for discussion and questions. And yeah, so this, as I said, this seminar touches on a number of different topics. So this particular piece is going to talk about gene expression and cell identity. All right, and I hope that this will play loudly enough, but let's see.
I'm ready.
Yeah.
So Laura Camel, the grad student that went and did a lot of these studies, she actually was trying to visualize RNA in the tissue. And she did that with RNA and C2 hybridization, which is shown here in black in these images. These are all coronal sections through the hypothalamus, and it's basically just showing you one side. Until now, I've been showing you two sides, but the sides are the same. And so I don't have to show you both. But you can see here that there are two genes. There's the TAC1 cluster on top and the reprimo cluster on the bottom. And the TAC1, again, is the one that we'd already defined. I had already shown that it was involved in movement. And you can see that there's more TAC1 expression in the VMHVL of females than there is in males, consistent with a female-specific effect on physical activity. But the gene on the bottom, reprimo, was super exciting because the sex difference was much larger. You can see that on the right, the female expresses a lot of reprimo in the VMHVL, and that it's pretty confined to the VMHVL, the area that we were really interested in, where the estrogen receptor is. And that on the male side, on the left, there's very little, if any, reprimo detected in that area. So we were really excited that this might be the population that we were looking for.
Okay, so basically here she's talking about two genes that her team was studying. They were expressed in the hypothalamus, and one of them is expressed in a sexually dimorphic pattern. It's expressed at a high level in females in this particular region of the hypothalamus, and not to very much, if at all, in the males. So how do you leverage this? Okay, so these are slides now from Shelley Tai at Glendale Community College, and just showing, you know, how she kind of leveraged this in her intro bio course, right? And so she built this into the classes where she's talking about how genes are regulated by tissue type and by cell type. And so, for instance, you know, she'll talk about how there are genes that are expressed, for instance, in digestive tissues, but not in other cell types like skin or in the eye. Okay, but then from the seminar, we've got these other genes that Dr. Correa talked about that follow a similar kind of cell-specific expression. All right, and so she then gets to talk a little bit about cell differentiation and how, as cells get specified into different cell types, you can identify the cell type in part by the genes that are expressed there, and how you detect that in situ hybridization is one way. All right, and then she's got a class where she talks about promoters and expression vectors, and gets to other parts of the seminar I'm not going to get into. So this is just an example of a syllabus where, you know, she'll have at the beginning, students watch the seminar in week 1, and then at different parts during the semester. So for instance, when she talks about gene expression, she gets into talking about in situ hybridization and, or no, maybe not in situ hybridization, immunofluorescence, immunostaining, and, the estrogen receptor and how they were able to localize estrogen receptor expression to particular neurons. Later on, she'll work in research deconstruction when she's talking about transcription and translation, and again, later on in sex cycle mitosis. So it's flexible. You get to decide where it fits best with your syllabus. We've had Faculty at Fresno State did this a little differently from the faculty at Glendale, and the faculty at Santa Monica did it differently from the other two. So each group gets to figure out how they want to incorporate it. So I'd like to take a few minutes for you guys to discuss amongst yourselves. How might you, if you, first of all, I guess I should ask, how many of you teach a biology class? Okay, all right. So, yeah.
Biological anthropology, it's kind of close.
Okay. All right, so I would have asked how you might fold this in. Let's maybe, it's a small crowd, let's just kind of go around the room for a second. So biological anthropology, and then mainly what else is it? Are there
I
other?
mean, I'm anthropologist, I teach in all
Anthropology.
fields, but
Okay,
I just think one thing I really love about this is just, flipping this in a way that's deconstructing is a different way of acquiring the language and concepts. And I think that, you know, rather than this consistent kind of like building concept after concept after concept, seeing it in motion, listening to it, and finding clarity in the dissonance is what students are starting to do by watching this in advance. That's just really smart. And I wondered how, just as you did this, the kind of more traditional mode of teaching is building scientific inquiry, you know, that's kind of stacked. How you balance that, because the deconstruction versus that kind of like forward thinking inquiry and no, that doesn't work, move into this. No, that doesn't work, move into this as you're teaching scientific methods. Just wondered if that's even a challenge as you deconstruct.
yeah, I mean, I think it'd be interesting to, so I have to confess, I have not actually tried to do it in the context of an intro bio syllabus. This is, Alex, I don't know if you could speak to this at all.
Yeah, the way we usually do it at SMC anyway is we'll take I don't know, 3 or 4 weeks to set up some basics, real basics, either with the non-majors or the majors. We'll just set up some basics, and then we'll show the video, and then we'll get going with the deconstruction after that. And it seems to work really, really well to be able to weave it into the syllabus and to, you know, show. I was thinking about it while she was talking, and I think about these, we don't often have time to just sit down and think about things. And so, I was thinking about how many times have I taught about Darwin's finches, for example, and that I can draw on Lisa Torres's bioluminescence story and talk about the radiation and speciation of ostracods in the ocean and how awesome it is to see the bioluminescence in the ocean is just so exciting. And so to be able to weave that kind of thing in and help students see how to think about science rather than just saying, here's how we think about science. We're showing them a pathway for them to think about science. Because in reality, each one of them comes from a different perspective, and they bring a whole new set of questions about science to the table. And that's the beauty of it, is to allow that to sort of bubble up through this instead of us telling them how to think, which is basically how I was taught. You know, and so I think we're giving them some background, weaving this in, allowing for that to bubble up, and also, you know, kind of guiding the pathway of, you know, well, that wouldn't work because of this, but this actually, let's build on that, you know? So, and to be able to bring something new to the table for me, like I've been teaching for 30 years, so do I have to talk about Darwin's finches again? You know what I mean? So I'm excited to bring it to the table as well. So it's that part's really, it's been really, really fun. And then just to add to this, which wasn't part of your question at all, is just to say, to see the students actually have enough confidence to get up in front of Stephanie Correa and Lisa Torres, and I didn't see the chemistry one, but I've been to many of the question and answer periods for both of them. that they have the courage to come up and just ask these really inquisitive questions about science and about their work is a really amazing thing. Like, they have transformed into scientific thinkers, which is so beautiful.
Yeah.
So,
Yeah, so touching on that last part, I've also eavesdropped on a number of these Q&A sessions. And I remember the first one with Glendale. We had a student in the class who asked a question. It was a really good question, scientific, about the science of the seminar itself. There were a lot of questions also that are really great that are about career path and how'd you get involved in this? How can I get involved in this? But there was this one student who asked a question, and Stephanie said, you know what, we've actually never asked that. It's a great idea, and we're going to do the experiment. And I could just imagine like how exciting that must have been for that student and for their classmates. And I emailed her after the Q&A and said, thanks so much for taking the time to do this Q&A. And she said, oh, I had a great time. And by the way, we really are going to do that experiment. I'm not just look. So it's like, here's this, student in first or second year student in a college classroom who's now made a contribution to a lab's research progress. And then the other one that comes to mind was at Santa Monica. You were there for this one. It was for Lisa Torres. This was general biology, and a student asked a question and said, I have never liked science, and I was never interested in it, and I never felt I was good at it at all. I never really cared about it until I studied this seminar. And now I want to know, I'm a public affairs major, I want to know, how can I weave science into my major, like in my career path? And I was like, right here's my, so that can happen. I think a big part of it also is you're providing context, right? It's no longer taught in a vacuum, but they can see how this applies to the real world. So we were going to ask you to kind of think about how you can fit something like this into your syllabus. I guess I'd ask, are there chemistry folks in the audience? We have the chemistry module, touches on a lot of redox chemistry, thermodynamics, kinetics, equilibria. All right, and then general biology is the other one. So I guess
can I just speak?
Oh, yeah, please.
Because we offer at Santa Monica College, we offer a lot of the introductory courses in a lot of different disciplines within the biological world. And so we have had discussions about the fact that Stephanie Perez's seminar is really applicable to physiology. And so the physiology students could really benefit from thermoregulation and things like that in this seminar, and deconstructing it in a way that the physiology students can use it. We've used Stephanie's seminar in majors biology for sure, but we also took Lisa's seminar and applied it to the majors after they did Stephanie's seminar, and they moved on to the capstone course that was ecology and evolution, so we could use Lisa's seminar. And so there's like I feel like there's really a boundless ways that you can use these kinds of tools in order to help students really see clearly into thinking about science in ways that they just haven't before.
Can I ask some questions?
Yeah, please.
So I was trying to understand, because first when you said you were doing that as an elective course and you had a lecture and then 7 classes to deconstruct.
Right, and that was half of the 10-week quarter.
Half of the 10. So I was wondering, when you brought it to the other courses like at community college and all, so was it a separate elective course or was it a part of already an existing course?
Well, before we got the funding from the Learning Lab, we actually did do a separate course at SMC. And the problem is it's not a requirement for transfer or for graduation or anything. And so recruiting students was a very difficult thing. So that's when we thought, well, maybe we should think about incorporating the practice into existing courses. And that's how we've done it, is just taking the syllabi that we already have and saying, okay, this module would go in here, this module would go in there. And then we can help build the concepts that we're already teaching with the content that is in the deconstruction.
Because I'm also seeing, when you say that, also seeing connection with cures, like, course-based undergraduate research experience, there's a lot of, I think, intersections.
Mhm.
And I'm not in biology or chemistry, I'm a mathematician, but I can see how I can take this model, because we have been exploring how to do some of these things, because mathematics is another area, very hard to make students do research at the early, level. So it's really interesting. So that last question was, when you ask them to make these videos, so were they given sort of like that this is going to be for this kind of class, so we want you to hit this kind of topic somehow incorporate into, so that when you start deconstructing them, it aligns better with your class? I was just wondering if it was.
We didn't ask them to speak about anything in particular, rather, we chose speakers whose research we knew.
Would align with.
We
I
touch
see.
on a
That
number
makes
of
sense, yeah.
topics and would intersect with the syllabus.
Yeah, and I would say particularly with the non-majors, we were hoping for something that was kind of wow.
Yeah,
Yes.
Wow factor, so the bioluminescence hit that right away. And then when we spoke to Lisa, we said, this is gonna be geared towards a non-majors level. And for that reason, we asked her to cut it back, because we thought non-majors might not be willing to sit through a 50-minute seminar at the PhD level. So we asked her to cut it down to 36 minutes or something like that, and it's actually a perfect length, and, yeah.
Thank you.
Mhm.
yeah, so, I think maybe I'll touch on just introduce our module, and then, so we've created a toolkit for each one of our modules. It's available on Canvas Commons. The chemistry one is still actually under construction, but should be up fairly soon. We have the introductory biology and general biology modules available. So if you just go to Canvas Commons and search for research deconstruction, you should be able to find our toolkits. And if you kind of open them up, you see that there's the research seminar video, there's just some instructor resources, and then all the various class tutorial videos, as well as sample syllabi and possibly sample exercises as well. So I guess maybe if you guys want, can have a little bit of discussion amongst yourselves about, or amongst all of us here, about just, is this something that you could see helping you in whatever discipline you teach? And what might be the benefits, but what might also be some of the challenges that you would anticipate in leveraging an approach like this?
Do you think we're a small enough group to just have a big group
Yeah.
discussion? If you don't mind, I'll go ahead and start. I'm curious about sort of the scalability. So I hear from you the value of having that Q&A session at the end. But in terms of folks who are not in courses where the couple videos that you have available would be relevant, like what's stopping me from finding a research talk on YouTube and using it to do the exact same thing?
Nothing.
Is there's
So
no reason
I
why
don't-
that wouldn't
If you find
work
it,
in any given course, right?
right,
It's just
right.
the philosophy of start with a fully executed research project.
Yeah.
Leave the topics throughout your syllabus. By the end of the course, they should have learned about the research methods that were used, about the concepts that are relevant to the course, et cetera, right?
Yeah,
Yeah,
Is
and
that
I
it?
would just add that you might even reach out to that speaker and
Yeah,
see if
exactly.
they could do a Q&A.
Exactly.
I have to say, like, when we reach out to these people for a Q&A, they're like, yeah, absolutely, tell me when. They're super into it, and so I think you might find that
Yeah.
wherever they are, they'd be willing to like log into a Zoom meeting and.
yeah, it's one of the great things about remote technologies now, if we can do that.
Right. Although
And
I
it's also,
really,
Oh, I'm sorry.
So I was just gonna say, I really love the idea that your speakers are coming from the region that you're in.
Mhm.
I think that there's also like a
value.
familiarity or a closeness to the, role model thing that's happening there too, that like, this is a person who might be from California or went to an institution that I'm thinking about transferring to, or is doing research at an institution that I'm thinking about transferring to. Excuse me, sorry, I'm getting over it. Anyway, yes, okay, but I'm not off base here. I could, in theory, apply this method to any course, using any talk. Any
Absolutely,
research.
yeah. And the other thing that I'd point out is, it's not essential that you deconstruct the entire seminar, right? So if you take a piece, you could show them the whole seminar if you want, or you could show them a piece. If you only have time to deconstruct part of it, as long as you do so in a way that they understand it, like, that has value, right? And in our standalone course at UCLA, that happens a lot. We will have a speaker who comes in with 75 slides and 3 stories in their seminar, and there's just no way we're going to unpack all of that, even in 7 classes. So we'll take like one of the stories and try to do a good job with that and say, we're not gonna get to the other 2.
And as part of the work that I do as the assessment person, I've actually interviewed a lot of the faculty who participated in this. And they've actually been able to kind of, now that they have a handle on how to deconstruct, they've talked about taking papers and deconstructing pieces of paper. So like, you can't find a talk, but you have this paper that you think would be really interesting to your students, you could find pieces of a paper that you could do. So I think you could apply it that way too. There's just not a great talk that you can find. Although after today's earlier session, there's lots of good YouTubers out there, so.
I'd also say that this is doing it with research, that's kind of the lens to, which we approach this, but say in engineering, like, this might be applied to not necessarily research and discovery, but application and development, right? And I think the same ideas could apply. So you had a question?
Oh, no, so I just wanted to, earlier you mentioned some professional challenges, so as I think about incorporating this into my classroom, I teach at a community college. There's a balance we have to strike for the students to learn the technique, learn to work with their hands, set up the apparatus, a safe manner, so forth, and think about the research, and there's value for both. So as I'm kind of anticipating incorporating this into my course, that's a challenge I anticipate, is striking that balance. We only have limited amount of time and so much curriculum to cover, and so I just wanted to kind of see what your thoughts were.
So our collaborator at Glendale, Shelley, she actually incorporated this into her lab. So that was one way, and then two of the instructors at Fresno State used the modules to be part of case studies that they've had already in the class. So there's lots of ways to apply it. They all apply a different number of the pieces of the module, so you don't have to do all 7 of the videos, you could do 2, and then kind of build. I think the iterative process of it was really helpful for faculty, because they'd kind of do, okay, I'll do 1 or 2, and then they were like, yeah, this is working, and then they'd add 1 or 2 more, and so over time, they were able to sort of really like flesh it all out, and it found that there was a little bit of an added bonus when students have more opportunities to engage in it, just based on some of our interviews with them.
I'm interested too, the last bit you were talking about the proximity, and so what's also so compelling is that their stories. And so I wondered how you incorporate that, because we've talked so much about belonging and race conscious equity and that kind of like work of transfer and seeing yourself in STEM fields especially. So how do you bring those stories in? Do they share their stories, or how they got to?
Well, that's why we recorded those videos for the career paths, and so those are available to the faculty.
And then
have
they
the
can
students
deploy it at particular moments throughout.
Yeah, you can, it's just a 5-minute video, so you can just assign it. You can show it in class, or you can assign it before class and have a reflective assignment or something like that. One of our faculty talked about how, she grew up in Southern California, was the daughter of immigrants, 1st-generation college student. And I think a lot of our students can identify with that.
And have you seen that increase, just out of curiosity, in your completion, or is the data just too small?
in terms of staying in STEM or.
Yeah, staying in STEM, completing, transferring.
We haven't had enough time to
Yeah.
really be able to see. So I think right now we could potentially, with some of our earlier cohorts of students, see the transfer piece, but because it is one intervention at one
Yeah.
point in time, if it's their first semester, it's gonna take several semesters before they transfer. I would love to be able to see downstream, like how students are doing, if they take it in their first chemistry course, how do they do in their second chemistry course?
Yeah.
I will say our colleague at Glendale found that, and this is anecdotal, I don't have, hard, fast data to substantiate this, but she had students in her biology course where she used the deconstruction and ones who didn't participate in her summer lab. And she said it was a huge difference, the folks who had taken the deconstruction, because they were already hitting the ground running with research. So when she provided, it was kind of like a journal club, the papers, they already had a lot of competency to be able to go, oh, okay, I understand those processes, I can ask them the questions I need to. And she said that she felt a huge difference in the way that they were able to engage with the material. But we just don't have the ability to really follow up with these students as long-term, because there's over 1,000 students that have had exposure to this at this point, so it's very hard. And it's sadly only one intervention, or if you're lucky at SMC, maybe two.
Yeah, and at Santa Monica College, we don't follow students as they go out the door, and so, that would be really nice to be able to follow where people end up.
So we do have survey data from the students, to sort of see how they felt about the experience and how much they feel like they've learned. We've also taken, like Iris said, the questions that they asked and kind of tried to use qualitative methods to pull out themes and trying to see the kinds of, like, the sophistication of some of the questions that they're asking. So we have some of their actual content that they're providing, their feelings on it, and then we have the instructor's perspective on how they've seen students change. So we feel like we kind of have some triangulation of our data to say, yeah, there's something happening here based on all these different points of data. But we're not really able to track at these other places quite the way my data brings me. Like, how are we such a great study? We can, or we can, there's some data at the back of the presentation. We can kind of show you what some of the students are saying. Okay. We do now. We're gonna, what, 40?
We've got about 10 minutes.
Oh, yeah.
Yeah.
So, we had some short-term, medium, and long-term outcomes. The short-term focusing kind of again on just growth and their understanding of research generally, and their skills and their sense of what their understanding of the science and the seminar. We also wanted to see if they're changed in some of their levels of competence and interest in the science, as well as to see if where they started in terms of growth mindset and see if they shifted more towards that growth mindset over time. Spoiler alert, they already are pretty high in the growth mindset, so we didn't see a lot of movement because they were already towards that side. And then understanding how science fits into everyday life, science and society, which was a particular interest for some of our non-major students who may not have seen, like Iris said, with our the example of the student in public affairs, like how does science fit into my everyday life? That's not what I'm studying. So we wanted to see students' opinions about that. And then how impactful the deconstruction materials were on the students in a couple of different areas. Again, understanding whether they would like to take more classes with deconstruction and things like that. So we did have pre-post surveys for the students that included open and closed-ended items. This was from January, so we had one more iteration of the spring of 2024 that are not going to be included in here. And then we did a whole slew of different types of analyses. I had a really wonderful postdoc who was helping with some of the qualitative. She's now a researcher at Berkeley. Very sad for us, but happy for her. And so we worked together to code our open-ended responses, as well as our interviews to try to get some good iterator reliability. This is just some of, again, the level of understanding. So we asked students to think about what % they understood at the beginning, after the first watch, and after the final watch. And we saw big shifts in their levels of understanding. It was kind of surprising at first to think, oh, well, how did our non-majors think that they understood about 60% of it? But then again, talking to, Alex and others who use that module, that we know that it maybe didn't have quite the same scientific, technical language some of our the major one had, or the chemistry one had. So there's still a significant shift, but maybe they started higher because the level of language was maybe not as jargony or things like that, which is not a bad thing. And then their level of interest, we did see a shift, to be more interested over time, which I think is great. If you want to read to me. And then we asked the students in an open-ended question, what was the most impactful component of the deconstruction? You see almost half said it was understanding the concepts and getting that content knowledge. And then there was others that talked about the research methods. And then what was great to see is that we had a fraction who also talked about the real world implications of the research, which we know from previous work that that's very impactful for students that have been typically excluded or marginalized in these fields, is that they want to see that connection to the real world. Why should I care about this? And I know I want to be a doctor because I want to help people. Well, this is how we can see how this impacts our everyday life. So that was great to see as well. And then it's not on this. Next one. Okay, so I will say, since we put this together, my postdoc recently got my stuff back to me on Friday. So some of the questions that we coded 150 of those as a sample of those questions that students were asking, and the bulk of them were, technical clarification questions in the Q&A sessions, questions about scientific content, application of the research. A large portion of them, about 30%, asked about the social impact of that work. Another 30% was looking at research process, but then we see, again, maybe like 5% are asking about those researchers' personal experiences. So they actually, one of their questions, instead of working on the content, they found it meaningful enough to ask them, like, what persuaded you to go into this? And why did you choose this? And how did that, your experience with your family and things like that. So we see that there is sort of a wide variety of the kinds of questions that they're asking at the end of the Q&A.
At our last Q&A, one of the students in marine biology asked Lisa Torres about the bioluminescence seminar, if she believed in aliens. And there were a couple of giggles in the classroom, but she dealt with it with such grace. And so it was just such a nice way to, just make that student still feel like it was a valid question, and yet kind of dismiss that whole concept that has been coming around, that octopuses come from outer space. And so she did a very good job with that.
So, I just want to acknowledge folks. So, research deconstruction kind of grew out of our biomedical research minor at UCLA, and Paul Banerjee was a big driving force behind this and really going, along with Raphael Leno, who's part of our team here. I want to acknowledge all the various members of biomedical research minor over the years. Our partners at Santa Monica, including, of course, Alex, Bill Persuli, Darren Bechdesarian, and then some of these folks like Rashida Hawk and Kashante Johnson were involved in that earlier iteration where we just had an elective course that kind of inspired us to really develop this project. And I want to acknowledge Shelley and her colleagues at Glendale, and Chris and his colleagues at Fresno State. And of course, our assessment team, Mark Levis Fitzgerald, Casey, Britt Tobin Lindsay, have really been kind of driving all the data collection for this project. Okay. Yes, thank you.
So I'm wondering if you are thinking about or open to maybe do this for other areas like mathematics or, applied math or computer science, or because I feel like it could be a really meaningful experience for a lot of the students that we are teaching at CSU, because that's why a lot of us are experimenting with cures in our early courses, in our capstone courses. And I think I completely see that connection when you say they were asking a lot of personal connection and story questions, because I think the students that we are teaching especially I teach at the CSU, I think those makes the huge impact on them, what they want to do next, because a lot of them don't even know what opportunities they have or possibilities they have. So when they see someone like them talking about their journey, it's like, oh, that is a possibility for me, and so I think.
One of our speakers
Very
actually spoke about a nonlinear path to getting,
Yeah.
you know, there seems like, oh, you need a bachelor's and go to master's, you get a PhD, and you put that, and then you get a faculty job. And that was not her experience, and so to see that, but she still got to to that point of being a professor, teaching and doing something meaningful. I think it's very impactful for this,
Very, very
like, oh, you don't have to do it this way, you can have this different route and you can still get to the same place, that's great.
Yeah, Kimberly Tanner, San Francisco State, I want to say like the foregrounding of the research deconstruction seems like a real strength of the project. There are other approaches in classroom scientist spotlights that foreground the social story, and that don't go into nearly the technical depth, and are available across a lot of disciplines. So I think that I would predict that you just get different outcomes. So the outcomes that get measured for scientist spotlights are more around sense of belonging, perceptions of scientists. You're not measuring something that's about a research technique, so it depends on your goals. But there are other resources that are complementary to this approach.
Yeah, I mean, one thing we have seen, I think, with some of our data collection is increases in science confidence in students starting from a point where they hear this seminar, they don't understand a word of it. And then by the end of the semester, they actually understand a decent amount of it. And so they I think that it has contributed to confidence and, you know, possibly science identity as well.
But I also
but-
think like normalizing that, that's
Yeah.
normal to come into something new that
Yeah.
you don't know, and that's okay, because that's part of being a scientist is having those questions and kind of learning through, and it's okay that you don't know everything right away.
Absolutely.
Because
Yeah.
sometimes I think students feel alone, and I think everyone doesn't understand this, no, it's okay, we'll get you there, and I think it builds trust also with the instructor, thinking about today, like how do we build that trust, oh, it's okay, we'll help you get there.
Yeah. With respect to other disciplines, I absolutely think that it can work. That's the kind of thing we'd have to do through collaboration because, you know, we're biologists and I wouldn't trust myself to be able to like
Break down a
deconstruct
dam.
something and in number theory or something, but I absolutely think that the approach can work and you know, there might be some discipline specific tweaks that you
Yeah,
make because
absolutely, yeah.
of how, you know, how the work is done.
Yeah.
So,
The last slide.
yeah, so, happy to continue this discussion beyond, but before I let you go, our funders, have, kind of a, you know, survey they'd like you to participate. It's very short, few questions. So if you open up your Whova app and just go to, the agenda button on the bottom and then select our workshop. And then, click on, provide feedback, I think it is, or session feedback. They'd be happy, we'd be happy, thank
Oh,
you. And I think there may be like.
And you
or
can
something.
win a raffle prize.
Yeah,
The deconstruction materials are, for the two biology ones are available free on Canvas Commons. You can download all of it and kind of see what it looks like, and that way you have a sort of a template of what you could do with it. Not that you necessarily have to use that content specifically, but it is there, and you can see different ways
Yeah.
that folks have done it, and it will live on Canvas Commons.
Yeah.
As long as you have a Canvas.
and if you are thinking about trying out something like this, you know, you wanna reach out to me and just consult, I'm happy to, you know, advise and, or, you know, talk about your ideas about implementing it in your discipline. I actually have a colleague, at a college here in LA who is planning on trying this in math. So we'll see, you know, I'm sure it'll go great and we'll see how, you know, what we
Yeah,
have
I
to do to
can
make that
totally
work.
see that your model could work totally for
Yeah,
math.
forever. Yeah.
We just, and in fact, some of your guided questions, you know, I think I saw somewhere in the beginning, could actually
Yeah.
transfer over to different
That's great.
areas.
That's good to
so,
hear, yeah.
Yeah, and are your slides available on the app?
They are not, but I'm gonna have to talk to the technical folks about how to make them available.
Okay,
Because
that would be helpful.
that was our intention,
Mhm.
but this particular presentation was like almost 190 megabytes.
Oh,
And so we couldn't upload it,
oh,
so,
because of the videos, I think.
yeah, so maybe if we make a PDF.
Yeah,
We might be able
that would
to
be, yeah.
get it small enough that we can bring it up there. Yeah. Bruce. All right, well, thank you very much.
so much.
Appreciate it.
Such a cool idea.
Yeah.
Have you thought about doing it in K-12?
Right? It's like a mystery.
We've talked
and then
about
figure
it,
out.
but we haven't tried it.
It is.
I think it would be very fun to do for high school students, and then going like earlier than high school, obviously.
It would just be a different kind of video, but I think you could do
Yeah.
the same idea.
You're not gonna start with
Really
Stephanie
powerful
Correa's
things,
research,
right?
No,
but yeah, like,
Research is research, right? So this is all like anecdotal, but I have a middle schooler and a high schooler, and always my daughter is doing her science homework and she's doing homework. I saw part of the homework, and I'm like, no, but this is really cool because this is what scientists do. This is why
Yeah.
they have you do these things. It's because this is what scientists do. This is how it relates to scientific inquiry, and she's like,
Yeah.
what? I'm like, how are we getting our kids through the science curriculum without them? We're never telling them how it actually relates to what scientists do.
yeah.
And this would be a really powerful way
I
to be making those
there's a lot of
connections
different
between
senses
like,
about what scientists do.
Yeah.
we teach them the research method, we teach them the biological concepts, et cetera, and we have
Pure
the
kind of thing,
how
how
biologists use it.
Yeah,
As-
and too often
So what-
science is taught as the fact that you have to memorize. it.
Exactly, and not
And
a
that's not at all,
thing.
yeah,
Yeah.
yeah.
And I honestly feel the same way about math. I love math because it teaches you to think in a logical and sequential way and how to break down complex problems, and like, it's such a great way to think, and my daughter just like, what are you talking about? But I feel like this could be a really cool method to get in a more concrete way at here's how what you're
Yeah.
learning actually relates to what it means to be a scientist.
I think
Mathematician or someone who thinks mathematically.
it would be a lot of fun to try to, like, develop this for K through 8.
Right.
Yeah.
And you
Yeah.
were at the previous, like, our utility lab, where it's just,
Yeah.
this is that's all it is, it's all.
Yeah. With respect to other disciplines also, I'll say that until this project, we've never tried it with chemistry. And I was amazed at how well, now maybe we should do this particular seminar, but
Yeah.
the alignment between the chemistry seminar and the chemistry syllabus is nearly perfect, at least for that, general chemistry course.
Yeah.
Between, it was really impressive. Take a look at kinetics, thermodynamics, equilibria, acid-base chemistry, redox chemistry. It was like, you could just pick off every single part of the syllabus and it aligned with something from the syllabus. Thank you very much. Great idea.